Okay, so this is the fifth class. I apologize that I'm so late in this. I just got the time. So in this case, we are discussing immunodeficiencies. Okay, this just means that the body is not able to fight the immune system, and this may be due to the fact that antibodies are not working, complement system is not working. So it can be a combined reason that cells, antibodies, and complement are not working. Phagocytosis is not functional. Many reasons. So let's dive into it. Uh, so the first presentation just tells us that we have two types of immunodeficiencies, and they're primary and secondary. And primary, the main cause of it is congenital, passed on from the parents, genetic causes. The second one is acquired, is because of maybe the person gets infected with some sort of disease and causes them to be immunodeficient, and that's secondary immunodeficiency. And I'll show you in what cases exactly we can see primary and secondary immunodeficiencies. And the result, of course, is that we have more infections, and we have incidences where we can have autoimmune diseases and lymphoproliferative disorders or even malignancies. Okay, so let's just dive into the first one, primary immunodeficiency. In this case, we've got the reason for it is being congenital, and it's genetically determined. And so in this case, this can affect so many parts of the immune system, including the structure of cells, their maturation in the thymus or in the bone marrow, their differentiation, whether they're able to do specific tasks such as being a T helper or a cytotoxic T cell, and their function within organs and the immune system. Okay, so primary immunodeficiency is just genital and can affect many areas within the lifestyle of an immune cell. Okay, the first thing that we have to focus on, and just before I go on, this presentation is the first one among all the others, which needs so much memorization from your part. And so there are so many things that we need to memorize within this presentation, and I'll, of course, point them out accordingly. So the first part that we'll face in really actually needing to know or memorize things is here. And so in case of children, primary immunodeficiency is suspected in case of, let's say, 10 reasons, but from those I would memorize the first 4. And I got like a quick way to remember things. So in this case, we see 4, 2, 2, 2, and that's just something that you can remember. 4 is for ear infection, then 2 for sinus, then 2 for antibiotics taken orally, and then 2 for pneumonia. And then this is just a way to just remember what 4222 refers to. And of course, all of them happen within a year. So if we have 4 ear infections or more per 1 year, is an indication that we have a primary immunodeficiency in this child. Okay, the second thing is that 2 or more sinuses per year. This is an indication that we have primary immunodeficiency in this child, and then so on. For antibiotics, we're taking 2 or more months of antibiotic treatment, but it's not working. So this is an indication of PID. And finally, 2 or more pneumonias within 1 year for a child. This just tells us that there is PID. Okay, 4222. Second one is talking about adults. In this case, we don't talk about antibiotics, but just ear infections, the sinuses, and the pneumonia. And in this case, it's 221. Okay, and same period, 1 year. But in pneumonia, it just says that you have a pneumonia 1 year, one pneumonia in 1 year for the next consecutive 3 years. Okay, so let's say for 2020, I have pneumonia, 2021, then 2022. This is a sign of having a PID. Just memorize those. Okay, so that's the second slide you have to actually memorize. Third slide to memorize things in. So primary immunodeficiency is made up of 4 very general parts. And let's say primary immunodeficiency can be antibody immunodeficiencies, combined immunodeficiencies, phagocytes immunodeficiencies, and complement immunodeficiencies. And these four things you have to memorize, knowing that the most occurring one is the antibody, and the least is the complement, and so the combined and phagocytes come in between, okay, as the percentages here are stating. Okay, so this slide can be very confusing, but what we're looking at is basically the names of the diseases that lie within the four categories that we have just memorized. So again, we will have to memorize specific diseases that lie within the specific types of PIDs. Okay, so first of all, let's go with, let's say, combined immunodeficiencies without syndromic features. Okay, and that's number one. Okay, the second one is combined immunodeficiencies with associated or syndromic features. And so those two types lie within this one right here, combined immunodeficiencies. Okay, طيب هلا first one, combined immunodeficiencies, we have many different examples that lie within here, and it's in a table down below. So I will point at point number one right here, and I will scroll to the part where you can actually memorize the diseases connected to this first point. Okay, just focus with me because I'm trying to help you navigate this presentation and get the sense of it. So combined immunodeficiencies without syndromic features are found in this table. We're going to scroll down. Again, here, combined immunodeficiencies. This is the table that you will be referring to that first point. And so you just have to memorize each and every single one here and how an immunodeficiency is resulting of that specific name, okay? Just an overall idea of all of them. Basically, there's a decrease in T cell, there's either an increase or decrease in B cell or an increase in B cell sometimes, and a reduced antibody. Okay, so they'll all talk about those three factors, and generally in all of them, there's a general decrease in everything. Okay. Again, so if we are asked about examples of the first point of primary immunodeficiencies, this is a table that you will go to. Okay, so just memorize them. Okay, and then you can go to the next point. So combined immunodeficiencies with syndromic features, and here we have 5: Viscott-Aldrich, ataxia, telangiectasia, Nejd-Megen syndrome, D-George anomaly, and Job syndrome. And these should be memorized. Okay, now, okay, so we talked about point number 2, and we've given examples either with non-syndromic or the one that has syndromic, and you guys know now where we can find them. The third point here in this slide that refers to the name of diseases that lie within the antibody part of the PID, hence point number 1 here, antibody immunodeficiencies, and they're also 5, and all of them refer to the deficiencies in some way about the presence of antibodies, and their names: Bruton A gamma globulinemia, common variable immunodeficiency, selective IgA deficiency, transient hypogamma globulinemia, and finally isolated IgG subclass deficiency. Just memorize those 5, and they lie within the antibody immunodeficiency of PID. And these ones make up the most common part of PID, 50%, as mentioned above. طيب, let's focus on the ones that we mentioned. So we finished antibody, combined, now phagocytes. And phagocytes, diseases related to it, they're basically 4, but here in this slide, 2 are present. First one is chronic granulomatous disease, and leukocyte adhesion deficiency. Okay, so I will scroll down again to a different table to show you where we can actually find the third and the fourth type. Okay, so now just focus, phagocyte, yes. We go down. Okay, and they're found here. So I told you the first one is chronic granulomatous disease. The second one is leukocyte adhesion deficiency. And then the fourth one is Hideaki Higashi and toll-like receptor signal defects. And these four make up the phagocyte immunodeficiency. Okay, just for each and every one, you really have to know how this disease causes a functional deficiency. Okay, for example, chronic granulomatous disease through the defectiveness in ROS. LAD1, type 1, B2, beta-2 integrins are not working, so there is no adhesion. Leukocyte adhesion deficiency type 2, there's a problem in endothelial E and P selectins, hence causes failure of leukocyte migrations into the tissue. Leukocyte adhesion deficiency number 3, there's this problem in the inside to outside signaling that cause a problem in the integrins and so hence allow for LAD to be produced. Here, Hideaki Higashi is basically when lysosomes and phagosomes are not able to fuse and hence not cause digestion of the phagocytosed bacterial body. Okay, toll-like receptor signal defects, of course, there is TLR and CD40 defects or signaling defects and cause defective type 1 interferon production. Okay, just memorize this and so you'll be good with the phagocytosis part of immunodeficiency. سكر سكر يا زلمه. اوكي طلعني معاك. سكر يا زلمه شرفك. اوكي. Now phagocytosis, phagocytes immunodeficiencies is done. So the first 3 points are good. Complement immunodeficiencies, they have not mentioned any examples about it. So just don't worry about it because I've solved some questions and sometimes you see those names and they ask you what type of primary immunodeficiency is, let's say, Viscott-Aldrich syndrome. So you'd have to select the option that says combined immunodeficiency. Okay, all good? All right, let's move forward. Now secondary, it's the acquired. How can we acquire secondary immunodeficiencies? Of course, by external factors. Okay, so for example, you consume a lot of alcohol, drugs, you have a lot of stress, malnutrition, you had a surgery, some treatment that you have taken such as glucocorticoids. Okay, what else you have mentioned here? Down syndrome, AIDS, right? HIV can cause an immunodeficiency for you and it can result in the secondary immunodeficiency, okay, not primary. In fetus sometimes or during the pregnancy period. Yes, so these are just examples of you to memorize for how we are able to acquire secondary immunodeficiencies. So now okay, so now we have PID and SID. Now when we have either of those, we are prone to very many infections, okay? And they can be either severe, persistent, they can be recurrent, or they can be unusual. And here, this table is just telling us that, oh, somebody has PID, somebody has SID. What infections can they be exposed to? For example, a serious infection that they can be exposed to is meningococcal septicemia. Okay, some other diseases that maybe they're exposed to, something that is persistent and is not being able to get treated by antibiotics, such as, let's say, the candidiasis, oral candidiasis. Yes, something that is unusual is the pneumocystis carini pneumonia, something that is recurrent, that is not able to that this patient is always having that problem. It always comes back to him, such as upper and lower respiratory tract infections. Just memorize those and you'll be fine. But I just wanted to get you to understand why they put those slides just like that. So we talked about PID, SID, and if, let's say, what causes them, and what type of diseases lie within the sections of those immunodeficiencies. And if you have those, what you can be exposed to. Some severe infections, some persistent infections, some unusual, and some recurrent. Here, again, memorization. My advice to you is maybe to write this division of cell arrangement on a paper maybe 2 or 3 times, and memorize at each stage what type of immunodeficiency disease you can get exposed to. Just quickly, stem cell. The stem cell can be defective, and a disease that can affect it immunodeficiency wise is the reticular dysgenesis. Okay. Now, the stem cell gives 2 types of cells, lymphoid progenitor cell and myeloid progenitor cell. The lymphoid progenitor cell give rise to pre-B cell and pre-T cell. And so pre-B and pre-T would give rise to, let's say, mature B and mature T. And then T cells can do their function, and then B cells can give even rise to plasma and memory. Okay. Myeloid, on the other hand, gives rise to neutrophils and monocytes. And at each stage, there is a immunodeficiency disease that affects that stage specifically. Again, lymphoid progenitor cell, we can say that a severe combined immunodeficiency can affect the lymphoid progenitor cell, so it's not able to produce pre-B cell or pre-T cell. Now, if pre-B cell and T cell were to be produced and the lymphoid progenitor cell is okay, severe immunodeficiency, combined severe immunodeficiency can affect those 2 cells and not let them produce mature B cells and T cells. Okay, one more thing that can affect the pre-B cell to produce mature B cells is the X-linked A gamma globulinemia. Okay, and pre-T cell going to get mature in the thymus can be exposed to a deficiency in its development, and this disease that causes that is called DGeorge right here. And then finally, if the lymphocyte is produced, which is T cell, and let's say it's like a full functional T cell, it can still have a defect of having bare lymphocyte syndrome, in which I think we don't have any antigens that can help it do its functions, or like receptors on its surface, so hence bare lymphocyte. Or it can be affected by the Viscott-Aldrich syndrome. Okay, so in that sense, you just write down this scheme and just fill in the diseases that can affect every stage and try to memorize that, because I think they will get some questions about this. Okay. This is just a way in which we can identify whether the child has an immunodeficiency or not. And you can just read it for yourself. I'm not going to go through it, but the conclusion about it is that if a person has immunodeficiency, then they should not be given vaccines, because vaccines can induce a reaction within them that can make them sick because their immune system is not functional. Now, the immunodeficiency workup and some tests that we can perform to evaluate the presence of different cells and the functionality of them. This is something we have done already in different slides, the pictures that I've shown you before. It's just repetition. Now we're going to dive into the lab diagnostics, and for every cell, what can we do to figure them out? So let's go into specifically B cell evaluation. Now, if we want to know the numbers of B cells, now we have gone through this in the previous classes, but here we're just going through them again. I advise just memorize these slides very carefully, especially for, let's say, if they ask about a B cell evaluation and you have that snapshot of this presentation in your mind. So if let's say they ask for the B cell evaluation, the first thing we need to do is actually count the number of B cells present, right? And so in this case, we would count or be biased against all the other cells and chose only the B cells. And how we do this is by using, let's say, WBC's counts, And we are going to probably use some sort of antibody system that can be specific towards the antigens that are found on the B cells. Hence, we look for antigens such as CD19 and CD20. And you can memorize some more if you'd like, related to B cells. So CD19 and CD20. Also, we can go ahead and count for the antibodies that these B cells produce, and this will give us an idea whether the B cells are present or not. And so in this case, we will just check for the presence of the Ig different classes, such as IgG, IgA, IgM, IgD, IgE, or check for the presence of the subclasses, hence IgG1, IgG2, IgG3, IgG4, for IgA such as IgA1 and IgA2, and the others, as you know, we don't have any subclasses. So you just can count the presence of those immune cells and hence the presence of those antibodies. And hence, if you find, then this tells us that B cells are functional and they produce antibodies. Now, that's just for their presence, like counting their numbers and the numbers of the antibodies that they produce. We can also check if their functionality is working. And so there are 4 ways to do this, and just memorize what's written here. They're just basically tests that help you know whether these cells produce antibodies or not. And so first one is natural antibodies, and the tests isohemagglutinin and IgM antipolysaccharide antigens. These are things that can maybe provoke the activations of B cells and help them produce antibodies. The second way is protein antigens, and this is what we have learned. This is the thymus dependent response, and of course it produces IgG antibodies, in this case specifically 1 and 3. Polysaccharide antigens is in the presentation number 4, I think. Yes. It just mentioned sugar antigens instead of polysaccharide antigens, and this is the thymus independent way, and it produces IgM or Ig2. And finally, you can check for the function of the B cell in class switching analysis. IgE production happens here by the co-stimulation between IL-4 and CD40 ligand. Just memorize this very well, because maybe they'll just ask you a question like protein antigen. What's this test used to evaluate? You can just select B cell. Yes. In the same sense, T cell, NK cells. So T cell, white blood cell count. For T cells, of course, we're going to look at the antigens present on T cell surface, and in this case, it can be CD3, CD4, CD8, CD25, CD45, and in NK cell CD16 and CD56. Or Kir and Car family. And you can also look for granzymes and performance that are produced by these NK cells that help them perform their cytotoxic function. And finally, the functionality can be tested by delayed hypersensitivity test, lymphoid proliferative test that we have already studied before, activating those cells by mutagen and antigens, and then using maybe, let's say, CFSE or Burdu, or what it was called, tritium labeled thymidine. Or you can also test the cytotoxicity assays such as chromium, for example, or MTT or flow cytometry. This is the one. There's a table I told you about, talking about combined immunodeficiencies without syndromic features. Phagocytic cell assessment, easy. We took that before. White blood cell different count, again. Functionality, chemotaxis, absorption assessment, oxygen burst. Chemotaxis by using agarose gel and Boyden chamber. Absorption assessment is by labeling those bacteria with fluorescent material, and then testing what cells absorb this bacteria, or looking at one cell absorbing how many bacteria. Oxygen burst can be tested by 3 ways: NBT, chemiluminescence, and cytochrome C reduction. And we can also test for the expression of adhesion molecules on those cells. Now, what's this telling us? Phagocyte cell evaluation can be of spontaneous NBT test or stimulated. So spontaneous in this case means the cells are not activated, they're resting, but we induce activation. Okay, and so in this case, let's say, okay, no, we don't induce activation, but they're just resting, but they can still do some phagocytic-ish function. And so in normal cases, you will have 1 to 8, sorry, percentage of those cells having a positive NBT test, so that they're like kind of ROS active inside. Yes. Now, if there's more than 14% active, this tells us that, okay, we're suspected of having an infection. Okay. On the other hand, an already activated phagocyte, okay, usually by bacterial endotoxins, let's say, usually what should happen is that 80 to 90% of them, they should be active and functioning to kill to kill that bacteria, right? But when we have a case where there's less than 40%, now we are very interested to know if there is a metabolic impaired phagocyting cell. Yes? So that's just the difference. Again, this is the phagocytosis part of PID. Complement evaluation, how we do it is by CH50 and AH50. CH50 is a complement testing, and AH50 is the also both of them are complement testing, but each one is different for different complement pathways. CH50 tests for classical complement activation system. AH50 tests for alternative complement activation system. Here, what they say is that for different diseases that we have, what do we do to cause treatment? For all of them, just remember, you give antibiotic prophylaxis and antifungal prophylaxis. The only two that have an additional thing is SCIDs, which we give antibodies, and B-cell disorders, we give antibodies as well. This is just repetition, 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 and we're done. The next thing we're going to discuss is HIV. Where's HIV? No, that's one. Sorry. Holy crap. Okay. There. So HIV is a quick one, 12 slides. Oh, I think I need to share a different one. There. Okay, so I hope you guys see HIV. Yes. Okay, so HIV, scroll through that. What it does is basically a virus that contains viral RNA lying within a lipid membrane. On the lipid membrane, it contains receptors or signaling molecules called GP41 protein and GP120 protein. And these are the ones that help it shake hands with all of the cells that it affects. The cells, on the other hand, would contain also hands that help it shake hands with the GP41 and GP120 of the virus. So these things are receptors such as CD4. In this case, we're talking about what, let's say, for example, macrophages, CD4 T helper, CD4 memory, and let's say antigen presenting. Okay, and so CD4, and on top of that, they need co-stimulation molecules. In this case, let's say for macrophages, it is CCR5, and this is something that you need to memorize. For CD T helper, it is CCR3 and CXC4, and for CD4 memory, it's CCR4, I think. CCR4. No, here. So CCR5, yes. So CCR5 macrophages, CXCR4, CCR3 for CD4 T helper, and memory CD4 contains CCR5, and APC, I don't know, it's not mentioned here, but these are the 4 cells that are susceptible to HIV infection. So what I'm looking at is HIV, and what I'm looking at here are the cells that can be susceptible to be infected by HIV. Like the first thing is what they shake hands by the things that we just mentioned. Now, the viral RNA goes inside. The first thing that happens, that the virus uses the cell's reverse transcriptase enzyme to reverse the viral RNA to viral DNA. And then the virus uses the integrase enzyme to integrate its viral DNA into the cell's DNA, and then causes transcription of the whole created new DNA. And this causes the production of proteins or another viral RNA, either to pack that viral RNA into another ball to go affect other cells, or it can produce proteins. And so proteases are also needed to cleave those proteins and help this virus affect and cause drastic effects on our immune system. Well, how can it be transmitted? Sexually, handling blood, handling, let's say, medical equipment, and through pregnancy, mother to the child. the counts of all of the counts of the HIV within the bodily fluids can be found in the sperms, 10 to the power of 6, the most, then the blood, 10 to the power of 4, 10 to the power of 5, then it can be found in the saliva, about 1 copy, and then finally very low in the breast milk, sorry. And then finally, why is it very hard to fight the HIV? First is because it's able to change its surface antigens very well. So if the body produces a lot of antibodies towards that specific antigen, it can switch that antigen, and then it's so hard for us to actually fight it because it can always switch antigens and we'd have to always produce antibodies. In this case, it keeps surviving. Second way is that it is always inside of cells, so it's not exposed to the immune system, and so it will have, our immune system is not able to actually see it because it's always hiding within the cells that we have mentioned, such as macrophages, CD T helper cells, or CD for memory or antigen presenting. The third way is that it actually reduces the expression of MHC1 molecule. Why is that important? Is because cytotoxic T cell CD8 is not able to kill the cells that are infected by this virus, is because this virus decreases the amount of MHC1 on its surface, which are needed to bind to the CD8 cells and initiate cytotoxicity. If you're confused about NK cells, okay, you can then ask, okay, if cytotoxic T cells such as CD8 are not able to kill this HIV because of this expression of MHC1, why can't NK cells kill it instead when MHC1 is less expressed? Because if MHC1 is less expressed, NK cells can then be activated and not inhibited, and cytotoxicity of NK can work. Because HIV is also able to suppress NK functionality through different ways, not necessarily through MHC1 hiding. Okay. FAS-L, and this is where I'm coming to this next point, is that it is able to express the FAS ligand on its surface, and this is where it can actually kill the natural killer cell. Okay, so although a natural killer cell can activate cytotoxicity on cells that do not express MHC1, but the virus is also more clever that it expresses FAS ligand, which can bind to FAS receptors on NK cells and kill the cells. Instead of them killing the virus, it is able to actually kill our killing cells. Okay. The last thing is that it's latent, so it's hidden within those cells that are in our body, such as macrophages and T memory. So we cannot actually treat it, because even if you give treatment, after some time, this latent virus is able to activate itself and cause immunodeficiency. Next, this is just a graph to tell us what's happening, and to dive into what I was actually saying. The virus decreases so much here because antibodies in the yellow line increase, but it comes back to increase again because now it changes its antigens on its surface, and these antibodies in yellow are not functional anymore, so it comes back to becoming live again because these antibodies are not functioning anymore. That body should be able to produce new antibodies that fit the new antigen, and even if it does that, the virus goes back and changes its antigen again. How to diagnose HIV infection, and what do we do to test them? The first thing is that we need to measure number of our viral RNAs within the blood. Second thing we need to do is that we have to figure out what the second thing to know is how do we know that HIV is present? Is that maybe we can check for diseases that can happen, the opportunistic diseases that can happen in the presence of HIV. And in this case, it can be the toxoplasmosis of organs, it can be tuberculosis, it can be antifungal infections, it can be the pneumocystis jirovecii pneumonia. And those four are very characteristic of the presence of HIV because they are opportunistic to its presence. And the third thing that we have to check is the possibility of cancers, and these cancers are very specific to HIV, such as Kaposi, Burkitt, and cervical. Tests. We have rapid tests, we have screening tests, we have confirmation tests, and we have genetic. Rapid is just a quick way to know if there's HIV or not. Screening is such as ELISA, confirmation such as Western blot, and finally genetic such as PCR. There's just a way to just diagnose HIV, and they follow the testing methods here. We get screening 2 times, and then we confirm it. Okay, just how to know HIV. treatment, and that's the last slide, I think. 2 times nucleoside reverse transcriptase inhibitor. Okay, why is this useful? Because as I said, when the virus is inside the cell, it uses the cell's reverse transcriptase. And so a good way to actually tackle HIV is to give treatment of reverse transcriptase inhibitors. And in this case, 2 times nucleoside reverse transcriptase inhibitor. This 2 times is very useful because I've seen it as an answer in the questions. Actually, I don't know what 2 times is. And you just have to memorize the names in here. So I just had a very quick mnemonic: late, Late, lamivudine, abacavir, tenofovir, and emtricitabine. And plus, you can use this plus a protease inhibitor, or this plus an integrase inhibitor, or this plus non-nucleoside reverse transcriptase inhibitor. And the second part have names as well, such as raltegravir or ritonavir, that boosts lopinavir. Okay, so just memorize this and you'll be done. This was class 5, and I hope I've noted down what you need to focus on and memorize. Just make sure that you have all of those diseases, all of those categories of PID, memorize some causes of secondary immunodeficiencies, the evaluation tests that are used to evaluate all of the cells present, and the treatment here. And then I think you'll be fine. And of course, for HIV, what are the things that are found on HIV surface that help it to interact with the 4 types of cells that I mentioned, what those 4 types of cells have on their surface that the virus uses to interact with them. Then reverse transcriptase, integrase inhibitor, and protease molecule, and protease enzyme as well, hence protease inhibitor. And protease enzyme was actually used to cut down or cleave the proteins produced so that it can be used to help the virus make its function happen. And what else? Yes, the screening tests, the diagnosis of HIV, why HIV is very hard to fight. Yes, and that's it. Okay. Good luck on your tests, and I hope those videos have been helpful. And I hope everyone passes.